Hello everyone, welcome back to my YouTube channel Biologycolony. In this video, we are going to discuss the model organism of subphylum Urochordata, Herdmania, its general features and external morphology. This topic is relevant for various examinations like RPSC, other state PSCs, GATE, BSc, MSc exams, TGT, PGT and many other similar examinations. Do subscribe for more such content. Herdmania or Rhabdocynthia, it is also known as sea squirt. Why is it called so? Because it contracts its body to squirt water simultaneously or independently through the atrial and branchial apertures. It was called as Rhabdocynthia, but later it was named after the discoverer called Herdman. So the classification is it belongs to phylum Chordata, subphylum Urochordata, class Acidiaceae and order Pleurogona. Habit and habitat. It is exclusively marine and it lives in shallow waters. It is solitary and sedentary, that is, it lives alone and fixed at a position. It is commensal with Turbinella pyrum, that is a gastropod mollusk. Commensal means it does not harm the Turbinella pyrum or Turbinella pyrum does not harm it, but it is getting advantage from the Turbinella pyrum as it resides inside its shell. So it is plus 0 relationship. Green sea algae commonly resides test of Herdmania. Ciliary feeder, it is ciliary feeder. How? Because its pharynx has various stigmata that acts like sieve so that it can filter out the large particles from the smaller one. So it is microphagous as it feeds on the small particles or organisms like diatoms, algae, crustaceans, mollusks, coelentrates and protozoa. Next we have it is hermaphrodite, that is both the sexes are present inside a single organism. It has external fertilization, that is the fertilization occurs in the water or the sea water as it is marine. So sperms and eggs both are released in the water, but here occurs cross fertilization as the timing of the release of the gametes is different. Egg is released before the sperm, so it is cross fertilization. Development is indirect. Egg is free swimming tadpole larva. Egg gives rise to free swimming tadpole larva. It has retrogressive metamorphosis, that is free swimming larva converts to sessile adult, that is the degenerative condition. Now let's talk about the external morphology. Well, its shape is rectangular, purse, bag or potato-like. It is laterally compressed. Its size is about 9.5 cm by 7 cm by 4 cm and its foot is 3 to 4 cm long. Its color appears pinkish due to superficial blood capillaries present on the test and bright red patches present due to the vascular ampullae that are found on test. Division of body is body proper and foot. Body proper contains branchial and atrial siphons. It is broader and longer. These siphons have various openings or the two openings called branchial aperture, that is also known as mouth or incurrent opening, and atrial aperture, that is the excurrent opening. Influx of water carrying food and oxygen etc. occurs through the branchial aperture and exists, and we have outer tunic or test present. We have foot region that is made up of entirely by test or tunic. It appears dirty due to sand particles, shell pieces and other foreign materials that are caught up or trapped inside it. It is useful in attachment. When Herdmania needs to attach to the hard substratum, foot is absent and body proper directly attaches by forming a broad, flat and concave base. When there is a sandy bottom, foot extends, which may vary in shape and attain length of about 3 to 4 cm. Foot acts as a balancer. It is completely made up of test. So as you can see here in this diagram, It represents Herdmania pallida species. It has anterior side and opposite it has posterior side, dorsal side, opposite is ventral side. Anterior side consists of branchial aperture or mouth, which leads to the branchial siphon, then pharynx, and all the internal organs are present somewhat here. Then anus opens into the atrial aperture through the atrial siphon at the dorsal side. This is the body proper, and it is covered with the test layer or tunic. So um, next we have posterior foot, and it is completely made up of test or tunic. It catches the foreign particles, that is seashells, etc. Now let's talk about the test or tunic layer. It is a protective jacket or covering. It is soft. It is 4 to 8 millimeter thick and leathery. It has or it acts as accessory respiratory organ and a receptor organ due to the presence of ampullae. As you can see in this diagram, these are the test vessels, and here there is a knob-like structure that is called as the vascular ampullae. It provides red patches to the test, and it acts as a receptor organ as it is composed of or it is associated with the nerve cells attached to it. So the test layer is translucent while in young it is transparent. It is wrinkled in appearance due to the folds and depressions that are found on its surface. It is secreted continuously by epidermis of the mantle, that is the body wall. It inhabits algae, hydroids, anemones, mollusks, barnacles, and various non-acidians. It is composed of matrix. Here is the matrix, that is the gelatinous matrix made up of polysaccharide tunisin that is similar to cellulose C6H10O5N. It consists of various cells, corpuscles, that is mesodermal in origin. It has large eosinophilous cells that are spherical and stained bright red with eosin. Small eosinophilous cells, eccentric nucleus is present inside it. Small amoeboid cells, that these are small cells with amoeboid nature, that is they are irregular in shape or have no shape. Then we have spherical vacuolated cells. These are small and possess vacuoles, as you can see in this diagram. Next we have various other cells which we'll discuss in the next slide. In this diagram you can see here we have megasclera and microscleras, and these are the types of spicules that are present on the test. So the next cell or corpuscle is granular receptor cells. It is sensory in function, surrounded by nerve fibers. Next is small nerve cells. These are branched, as you can see in this diagram. They have 2 to 6 long dendrites, and they are present more in number. They play an important role in communication stimuli from receptor cells to the other part of the test. Then we have squamous epithelial cells. These are the hexagonal cells found on the test. Next we have interlacing fibrils. They form a fine network like smooth muscle fiber or nerve fibers. Then we have blood vessels. We have a network of vessels that are found in Herdmania. Ampullae is present, that is the terminal knob-like structure or pear or oval shaped. It is responsible for the appearance of red patches, and it serves as accessory respiratory as well as receptor organs being connected to nerve cells. Then we have spicules. These are the large number of calcareous spicules with numerous spines all facing in one direction. As you can see in this diagram, these are the type of megascleras. Megascleras are large in size and present everywhere except heart. They are of two varieties, that is one is pipette shaped, one is spindle shaped. So the pipette shaped one have a spherical swelling in the middle with two pointed ends on each side, and they resemble a pipette that is used in various laboratories. Then we have spindle shaped. These are more abundant in number and present in bundles. It has 20 to 60 equally spaced ring of spines. Next is microscleras. These are very small, 40 to 80 micron long, and these are knob-like head with tapering body. It has ring of spines. These are found only in test region. Spicules provide an internal rigid supporting network or framework like endoskeleton. They keep the mantle firmly fixed to the test, stiffen the walls of blood vessels to avoid their collapse and ward off the predators. So these are the function of the spicules. Next we have mantle or body layer that is present under the test, or it is encased by test which it itself secretes. We just studied that epidermis layer of mantle secretes the test or tunic layer. So this mantle is suspended inside the test and attached only at branchial and atrial apertures. So it appears bag-like. It is thick, highly muscular, opaque on anterior, thin, transparent and almost without muscles at the posterior region. Then we have visible internal organs are found that are the viscera, and mantle encloses a large water-filled cavity, the atrium. As the Herdmania lack coelom, they have a cavity that is called the atrial cavity or the atrium. Then mantle layer is composed of three cellular layers, that is outer epidermis which secretes test and made up of single layer of flat hexagonal cells. Then we have mesenchyme layer. It lies below outer epidermis and is derived from mesoderm. It contains connective tissue fibers traversed by nerve fibers, muscle fibers, and extensive blood sinuses. Muscle fibers are unstriated and arranged in three sets: annular muscles, longitudinal muscles, and branchiolateral muscles. Well, the annular muscles surround siphon and they are circular rings and they cause contraction of the siphon. As you can see in this diagram, these are the circular ring-like muscles present at the branchial aperture as well as the atrial siphon. Then we have longitudinal muscles. They start from the branchial and atrial apertures and mediate beneath the annular muscles to the middle of the body on each side, and they cause the contraction of the whole body. Then we have branchiolateral muscles. They extend deeper between the two siphons. Somewhat here, there will be branchiolateral muscles. Then we have inner epidermis. It is ectodermal single layer of flat polygonal cells lining the atrial cavity. All the soft internal or visceral organs are present inside the body proper, enclosed within the somewhat transparent body wall or mantle. Then last slide is coelom and atrium. Well, as I mentioned earlier, coelom is greatly reduced and it can be found confined in pericardial cavity, gonads and neural glands. It has a spacious cavity that lies between the mantle and the pharynx, enclosing the visceral organs, that is called as the atrium. It is lined by inner epidermis of mantle. It is continuous throughout the body except in anterior and ventral regions where pharynx and mantle become fused. They have peribranchial cavity. It is a part of atrium that is narrow on two lateral sides of pharynx, and it communicates with cavity of pharynx through stigmata in the wall of the pharynx. As we'll study in the next video, that stigmata and peribranchial cavity all will be mentioned in the next video. Then we have cloaca, that is wide atrial cavity above pharynx into which anus and gonopores open. Then we have atrial aperture or atriopore. It leads cloaca to exterior through atrial siphon. Then we have sphincter muscles. It is present around the base of atrial siphon and they are strong muscles that regulate opening and closing. Then last we have atrial tentacles. These are the ring of slightly serrated processes that project into the siphon. Thank you so much for watching this video. Like, comment and subscribe. Biology Colony, and stay tuned for more.